Hello, guys. Welcome to another podcast with Martin Barna and Institute of Modern Nutrition. Today, we have a very special guest, and it is Gabrielle Fundaro, Dr. Gabrielle Fundaro. You may also know her as VitaminPhD on Instagram. Hello, Dr. Fundaro. How are you today?
I'm good. How are you doing?
I'm doing great. About some intro. I'm Martin Barna, online nutrition and fitness, and I will be your host for this podcast. Later on, the recording will be released on Spotify, YouTube, and Facebook. You can find all the links on the martinbarna.cz. Many thanks to Institute of Modern Nutrition, Miloslav and Lukas, for their cooperation and shared enthusiasm for spreading science-based information from the best in the world. Something about you, Gabrielle, a little intro. Dr. Gabrielle Fundaro, who you may also know her as her nickname on Instagram, VitaminPhD. For her clients, Dr. Fundaro combines knowledge in the field of nutrition with motivational techniques to support internal motivation and behavior changes for long-term changes and healthy lifestyle. She believes in the importance of plant-based diet combined with regular physical activity that we enjoy, and also in cognitive exercise as part of long-term sustainable lifestyle free from chronic dieting. In her work with clients with digestive problems, Dr. Fundaro uses a practical and personalized approach based on scientifically based information to adjust diet and lifestyle factors, which subsequently contribute to improving digestive comfort. She teaches courses in nutrition in the context of physical performance and health, physiology, and metabolism of life. Furthermore, Dr. Fundaro also brings science-based information to international seminars, podcasts such as this, and also contributes to magazines such as Shape or Oxygen. Dr. Fundaro is former associate professor at Georgia Gwinnett College and holds a PhD in human nutrition, food, and exercise. She also holds a bachelor's degree in exercise, sports, and health education from Radford University. In her spare time, she enjoys walking, reading books, powerlifting, and also enjoys spending time with her dogs, which are quite loud.
Yes. Hopefully
Did I miss something, Gabriel?
What was that?
Yeah, did I miss something about you? Is it correct?
No, that was great. You gave me a little bit extra credit on my position when I was teaching. I was an assistant professor, so one level below. But
okay, sorry for that.
Yeah, I went the route of entrepreneurship rather than going up for that next level, and no regrets. It's been great.
But still, you made many things in your life so far. I can see.
Yeah, when you put the list out like that, it's kind of surprising to hear it all.
Okay, can we start with questions?
Yeah, absolutely.
Okay, perfect. Okay, first question, because we will talk much about gut health today. What is the definition of gut health?
That's a great question that is challenging to answer because there isn't yet a consensus definition of gut health. We can think of gut health as sort of the lay person's term or sometimes even a marketing term. And so it's not one that's really used in the research world yet, because most of the time when we're looking at the gut microbiome in research, we're usually looking at just the gut microbiome, or we're looking perhaps at the relationship between the gut microbiome and a specific disease or a specific outcome after, say, a nutrition intervention. But I get this question a lot, and I have for a few years now. And so I've come up with sort of a working definition of gut health that I think addresses all of the aspects that people are thinking of when they say gut health. And I call it the 3 Ds of gut health. So we have diversity. So this refers to the actual gut microbiome. So that's the collection of microorganisms and all of the genetic material that reside in the gastrointestinal tract. And when we say diversity, we're talking about a wide variety of different microbes, and that there is a relative abundance, or there are proportions of the different strains and different microbes that maintains some level of what we would consider to be like a balance or a homeostasis. So that microbiome serves a number of different functions related to human health. So it serves an immune role, an immune function. So it helps to fend off potential pathogens. It also communicates with our own immune system. It also helps to harvest energy from the diet. And it likely also plays a role in just overall development of both our immune systems and even our neurological systems as we age. And there are probably other roles that we haven't uncovered yet. And so what we hope to see is a level of diversity that is sort of like, if you have like the Goldilocks story, you know, we have to have it just right, not too little and not too much. So we want to have enough that that ecosystem is still stable and resilient, but not so much that there isn't kind of like the predominance of neutral or beneficial microbes over the potential pathogens. And we also want to see diversity in terms of the different genes that are there. So genes are sort of like recipes for different proteins or different functions that an organism can perform. And so if we have a lot of different genes, because we have a wide variety of different microbes, that means that there's great functional capacity in the microbiome itself. So that's diversity. And then we have disease. So this is talking about the structure and function of the intestinal tract. So there is potentially, there's very likely a relationship between the microbiome and intestinal diseases and health, but we haven't established a cause and effect relationship yet. So that means that when we're looking at a disease process, we can look at that independently of the microbiome. And we might look for whether a person has a functional disease like irritable bowel syndrome, where the tissues look normal, but the function is abnormal, or an organic disease like ulcerative colitis, where the tissues are damaged, and that also has an effect on the function. And then we look at the third D, which is digestion. So this is more of the, we can look at it in terms of like the physiological process of digestion and absorption. How well is that intestinal tract producing digestive enzymes, and how well is it taking up the small monomers of the foods that we're eating? And then also just the subjective experience of digestion, which is more often what people are talking about. They're thinking about things like bloating, stool quality, constipation, gas, and what can often happen there is that people make assumptions about the overall health of the gut. So they think that like they can draw conclusions about the microbiome and potentially, you know, a disease or an impact on the like the lining of the intestinal tract based on the quality of their stool. And that's not necessarily the case. So for example, you might experience gas and bloating and feel uncomfortable after you eat something like broccoli or cauliflower, because the broccoli and cauliflower contain specific carbohydrates that those microbes can use for energy. And they ferment those carbohydrates and produce a lot of gas. Now that is a normal function of the microbes. And so there's not necessarily anything wrong with your gut health, but it can still be uncomfortable. So these three Ds are related, but again, we don't have a cause and effect relationship. And so it can be really difficult to come up with like actionable ways to impact gut health when we're looking at how many factors are at play there.
Mm-hmm. Mm-hmm. It's kind of interesting. It is not as simple as some people say.
Right. Yeah, exactly. Saying like good gut health or bad gut health is like saying, you know, we have a good ecosystem or a bad ecosystem. What does that actually mean?
Yeah, most times it's marketing when someone defines it, I think.
Yeah.
What are the worst gut microbiome facts that you've seen? Facts.
I think some of the most pervasive and misleading recommendations would be like saying that everyone needs to take a probiotic or that everyone needs to take any supplement related to the gut, because that's just not the case. That in some cases, the supplements just aren't substantiated by any evidence. But in other cases, like with probiotics, they do have specific applications, but not everyone needs to take a probiotic. And even if someone were to potentially benefit from supplementation, they would need to choose the correct probiotic for the job, so to speak. Another one that I think can be very misleading is this the fact that the majority of the body's serotonin is produced in the gut. It is true that we produce a great deal of gut-derived serotonin, but people then make the logical progression that it must mean that our gut-derived serotonin impacts our mood and can determine whether we're happy or not. But actually, gut-derived serotonin plays a role in intestinal motility, so the organized contractions of the intestinal tract, and also influences which substrates we might use for energy while we're fasting. But the gut-derived serotonin is produced via a slightly different pathway than the serotonin that's produced in our brain. And that final product, serotonin, actually can't cross the blood-brain barrier. So it doesn't act directly in the brain. So to say that, our happiness comes from our gut, or that mood disorders are caused by, imbalanced gut microbiome, really isn't supported by the evidence, and can be potentially misleading because people might forego certain therapies or pharmaceutical drugs because they believe that they can eat themselves into a regulated state. Another one would be, any recommendation about extreme dietary restrictions, based on food sensitivity tests, like an IgG food sensitivity test, or even something that's not based on any solid evidence, like the carnivore diet. A person might feel better coincidentally by eating a very restricted diet, but that doesn't necessarily mean that there are not risks long term. So I think, any type of information that happens to be misinformation or disinformation, which is a little bit more intentionally misleading, could be, potentially harmful. And so I would say those are kind of like the worst. That's the worst type of information when a person hasn't, done the due diligence in determining how accurate their statements are, and really evaluated the quality of evidence as well.
Very interesting. That one about serotonin is very widely spread. I think I've heard it many times, and to be honest, I did not understand it that well as before you discussed it like this. Also, today's favorite topic from listeners was the probiotics, right? So I think we will discuss this more. Okay, how can we positively affect gut health?
Well, when we think about gut health in its different parts, we can start with diversity. So what are some of the practices that might support a diverse, resilient microbiome? Based on both intervention studies, so where we take a group of people or maybe a group of rodents and we give them something like a dietary modification, or we have them exercise, or these larger observational studies when we look at a whole population of people, a few trends kind of stick out. So it looks like having a plant. When I say plant-based, I don't necessarily mean vegetarian or vegan, but a diet that is centered around plants and provides a wide variety of different plant-based foods. So fruits, vegetables, whole grains, legumes, nuts, and seeds. That's associated with increased microbial diversity. So people who have those types of dietary patterns tend to have higher levels of microbial diversity. We also see that people who have a high level of cardiovascular fitness and who do aerobic exercise regularly, or people who are even recreational athletes, tend to have higher levels of microbial diversity as well. So things like, eating a lot of different plant-based foods, fruits and vegetables and whole grains and such, and getting some physical activity in on a regular basis seem to be pretty strong influences on microbial diversity. But there are plenty of other factors that we can't really influence, like our biological sex, our age, our geographic location. We can change that, but the impact on the microbiome seems to be pretty strong in terms of kind of our country of origin. So the level, the magnitude of change that we can actually cause through things like diet and exercise are still relatively smaller than some of those more static factors. So just something to keep in mind. When we look at things like disease, there are some diseases that aren't necessarily preventable because there's a genetic component or we just don't know the cause. So things like IBS or inflammatory bowel disease, we have a predisposition and we really don't know what causes some people to develop those versus others. But something like colorectal cancer, we can prevent that, not completely, but there are practices that we can engage in that would reduce our relative risk of developing colorectal cancer. And again, that comes back to eating adequate dietary fiber and, from plant-based foods, and limiting the amount of processed meats that we are consuming. So things like sausage or like processed lunch meats. So, or like breakfast sausage. And then when we're looking at our digestive comfort, certainly, again, getting the right blend of soluble and insoluble fibers from a wide variety of fruits, vegetables, whole grains, and legumes, and adequate hydration as well to ensure that we're not having just a huge bolus of fiber and not hydrating adequately. And then also, if we are experiencing things like gas and bloating, just to kind of survey our dietary pattern and see, are we eating a lot of highly fermentable carbohydrates? And if so, doing some short-term reductions or eliminations and then reintroductions of some of the foods that might be the most obvious culprits. So something like garlic, onion, wheat, those tend to, and dairy, those tend to be pretty common, causes of some gas and bloating. Not to say that you eliminate them forever and never bring them back in, but you just kind of determine what amount is tolerable for you and your specific set of microbes, but ensuring that you're still providing them with adequate nutrient availability.
Interesting. And how does smoking and alcohol affect gut health?
The alcohol picture is kind of interesting because they have seen in some population studies that people who drink just a little bit of red wine each week, they tend to have a high level of microbial diversity. But there's this U-shaped, it's like a J-shaped curve that, if the more alcohol you drink, you're actually going to be seeing less microbial diversity. And we don't see that same, potential benefit with other forms of alcohol. There's not as much information about smoking, though.
Do we see the same benefits with just grapes, red wine grapes, or only with just wine?
So, yeah, exactly. So we do, but not necessarily. It could be for some of the same reasons. It could be that wine contains polyphenols, and then there's also polyphenols in the grapes. And we actually see that with a number of different fruits. So it could be grapes, it could be berries, or it could be vegetables as well. So these really, these colorful forms of produce provide these polyphenols, these plant-specific compounds that those microbes can metabolize and turn into something that's potentially beneficial for us. And it's another part of a diet that could be protective against colorectal cancer. But we're not exactly sure what it is about like the small amounts of alcohol versus, greater amounts, because you think, more alcohol means more polyphenols, but there's that negative impact of the alcohol itself. So don't go out and do probiotic wine or something.
Okay, good to know. There are many microbiome testings and food allergy tests, etc. Could we just explain to people which one works, which one don't work, or if any of those work, to be sure?
Sure. So I'll start with the food sensitivity test, since I mentioned that earlier. So a food sensitivity test claims to be able to identify foods that a person might be sensitive to. Now, food sensitivity is another marketing term, and usually people will say that, okay, if you eat this food, you might feel, gassy or bloated or have some brain fog or some fatigue. So they're coming up with symptoms, first of all, that are that don't have a known cause, and that everyone will experience at some point in time, because we live very busy, stressful lives, and we might not have super solid sleep habits or hydration and what have you. And then what they do is they measure in your blood the concentration of the IgG antibody. So antibodies are used by our immune system to identify and label potential intruders, to say like, this is an intruder, we need to mount an immune response against this. So the IgE antibody mediates the allergic response that most of us are familiar with, where we get like hives and our throat closes. The IgG antibody is actually a recognition antibody. So that says, hey, this is not an intruder. We don't need to mount an immune response against this particular protein. And so what you'll find is that when you do this test, you'll have high levels of IgG antibodies for foods that you eat frequently. Now, the test is valid in that it does measure IgG antibodies. It measures what it says it's supposed to measure, which is the IgG antibody. The inaccuracy comes from the application of that test, where a provider then says, oh, this means that you have a sensitivity to these foods and you should remove them from your diet because they're causing you digestive discomfort. They're causing these symptoms. You have a food sensitivity. That is not the case. Your body has just simply identified those, recognized them, and said, hey, don't mount an immune response. But what could happen when you remove those foods from your diet is that you remove a food that you could have an enzyme deficiency for. So say you're lactose intolerant. That means that you're not producing the lactase enzyme. And if you were to eat foods with lactose, like most dairy, then you would have GI distress. Or you're eating foods that are high in these fermentable carbohydrates. And so when you remove those foods, now you have less gas and bloating. But the answer isn't long-term elimination of these foods forever and ever so that you are left with only like 6 foods that you can eat. The answer is to do a systematic removal and then reintroduction of those foods to determine how much you can tolerate of each type of food. So those IgG food sensitivity tests are not appropriate. They're not being used appropriately. They're not clinically applicable. They're not clinically valid tests. So I recommend against using those or promoting them because they're highly misleading. Now, the other type of test that people often do would be a stool analysis, a comprehensive stool analysis. Now, it's not the same type that you would do if you were to, say, go to the emergency room because you've had chronic diarrhea for days and they're going to test you for C. difficile or for a parasitic infection. Those are clinically valid tests, and they are performed once a person has ticked off these boxes, of their other symptoms, like, all signs point to potential C. diff infection. And they will run tests that allow the provider to determine whether the C. diff that's present is actually virulent, is actually producing problematic levels of the toxins that can cause illness. Now, there are valid aspects to some of these comprehensive stool analysis tests, like they might measure something like fecal calprotectin, or I think that's the one that I've seen most often. So fecal calprotectin is a marker of potential for inflammatory bowel disease. And if that's completely off the charts, then it could be pointing to a person having a potential disease, and then they'd want to have a follow-up with a gastroenterologist. But for the most part, what people are looking at is they're looking at this long list of all of these different microbes that have been identified in their stool. And those microbes are associated with a specific reference range. Now, here's where it gets really tricky, because yes, the companies are using once again, it's a valid way of identifying microbes. They've taken that stool sample and they've either used an RNA-based method to identify bacteria or a DNA-based method to identify all known microbes. But for one thing, it's in a stool sample. That's not the same thing as actually testing the whole gut microbiome, because stool sample is going to be significantly different from the small intestine, and it's going to be significantly different from most of the large intestine. So it's only going to be representative of a small subgroup of microbes. Also, we're not able to actually identify every microbe that's present, because we haven't yet been able to identify all of them and give them all sort of like a barcode. That's the only way we can identify a microbe. We say, oh, it has we've identified this stretch of genetic code, and we can compare that to our reference library and say, oh, that's Staphylococcus aureus, whatever subspecies, whatever strain. But we haven't been able to do that for every gene, because there are hundreds of times more genes in the microbiome than there are in the human body. So it's much harder to identify who all is there. And the other tricky thing is that the reference ranges are set by the company. It's not the same thing as a reference range for like LDL cholesterol, because these reference ranges are based on just their population of healthy control individuals. So healthy, their healthy reference population, which might be 34 Caucasian females living in the United States. Well, what's considered to be a healthy microbiome still hasn't been established either, because healthy individuals in the United States will have a microbiome that looks significantly different from healthy individuals in Japan, who will look significantly different from healthy individuals in Germany. They're all healthy, but their microbiomes all look different. So how can we determine which group should serve as the healthy reference population for you if you were to go and get your microbiome sequenced? So then what happens is you get this report that says you have too much of this microbe and too few of these microbes, and you have this really high dysbiosis profile. And then a provider can say, well, you should take these, various probiotics to bring this group up, and, potentially like take this herbal antimicrobial to suppress this microbe, even though there's no evidence for that being the case. So it really becomes a tool to provide care that might not actually be necessary and really isn't based on solid scientific evidence that like this person even has a problem at all.
Mm-hmm. So from your point of view, when I have a client, let's say we think that some foods does not go well with him or with her, can I try elimination diet? Like you said, just one thing, let's say diary, let's say make it a week, then I can work with it like that, or should I send the person to the doctor? You know what I mean?
Mm-hmm. Yeah. My first recommendation for anyone that comes to me with GI distress is that they go to a gastroenterologist specifically, because if they go to an alternative practitioner, they might end up with one of these IgG food sensitivity tests or the comprehensive stool analysis. And really the goal is to rule out a potential disease, whether it's functional or organic, just to determine like, do you have IBS, IBD, or some other potential disease. Mm-hmm. And then once they are given a clean bill of health, then depending on their history, because again, there are risks with elimination diets if a person has a history of disordered eating, so if it looks like they're at potential risk, then it would need to be gone about, in a different way, perhaps, and perhaps referring them out to a dietitian who's, who specializes in that area. But if working with a person who has a clean bill of health, or is at low risk, then it would be okay for, to help guide them through, keeping that food and feelings diary to determine like, okay, is there a pattern after which, I eat this specific food and then several hours later I feel really crummy, like, or I get gas or whatever every time I eat this food. And then they can remove that food for a few days, see how they feel. If there's an improvement, bring the food back in. And if they feel really poorly and everything else has stayed the same, then it's likely that it is that food. It's still not a guarantee, but at least you've gone about that in a systematic way, rather than kind of just like taking absolutely everything out and leaving it out forever.
Yeah, of course. So you should be careful. I had an experience with client Wendy, she was called, and I did it the way you said. I've tried to eliminate, let's say, dairy, gluten, but separately. But meanwhile, I've always told her, Wendy, go to the doctor. Trust me, I'm a coach. Go to the doctor. It was 2 months, but she went to the doctor finally, and we found out it was a soy. I would never guessed the soy. Yeah. So it is good, a good example of why you should go to doctor, even though you have a coach and you are solving it. I'm not a specialist on this. I don't have the tools like a doctor. So I'm glad that I've sent her there, and I'm glad that you are confirming that I did a good thing.
Yeah, absolutely. And to that point as well, there are legitimate food allergens. And so a person could go to an allergist to be screened for a potential allergy. It's not the same thing as a diagnostic test when they do like the skin prick test. Again, it's a screening. You're looking at the potential there, but then there are subsequent steps like the elimination and then reintroduction, so they could determine whether it's a food allergy. And there are also non-IgE mediated food allergies that manifest a little bit differently. And so in some cases, they might need to do like an oral tolerance test. Basically, you eat it and then you see how you feel. So there are different ways of going about it that are evidence-based, but the IgG food sensitivity test is not one of them.
Yeah, okay. And I would just like to remind that a blood test doesn't work here. Is it right? The antigen blood test.
Yeah. So what they would want to do is like the skin prick test. That's so they basically like deliver a little bit of the potential allergen into the skin. And then if a person develops a rash or hives, again, that is a screening test. So they're able to determine like, okay, you could have an allergy to this, or in some cases, if you didn't react at all, you probably don't have an allergy to this. But yes, we wouldn't want to do the IgG food sensitivity test. That's a blood test, would not be appropriate.
Yeah, because so many people go to me and tell, I was on a blood work and I have allergy to this and this, that's why I'm asking, because I want them to hear it from you. Thank you. How about leaky gut? Is it a myth or a real condition? And does gluten cause this, if it exists?
So leaky gut, the term that I think is a little bit more, that's a little bit clearer physiologically, is intestinal permeability. And it does actually occur. It does exist. So this isn't completely made up. It happens when the intestinal cells that are in a single layer along the length of our intestinal tract, they're held together by specific proteins called tight junction proteins. And there are different factors that can reduce the integrity of those tight junction proteins, cause them to break down, cause them not to function appropriately. And so some space develops in between those intestinal cells. And then we end up with with leakage, you could think of it like that, of some contents from the intestines out into circulation. So we end up finding things that we wouldn't normally expect to see. Now, it could be something as large as an entire bacterium that is translocated, and that we see, that's associated with like sepsis. So in that case, this person might have ulcerative colitis that's really progressed and hasn't been managed. And so they have really large ulcers in their intestinal tract. It's not quite the same thing as leaky gut, but that's one way in which it can manifest, that we actually have large ulcers. But leaky gut, I think most of the time people are thinking about like it's permeability. There's just space between the intestinal cells. So there are factors that are associated with an increased likelihood of having altered intestinal permeability. So like more permeability than we would expect to see in a healthy person. That includes things like non-alcoholic fatty liver disease, inflammatory bowel disease, even acutely doing intense endurance sports, especially in the heat, and type 2 diabetes and obesity. So those are all associated with markers of increased intestinal permeability. So it might be that we measured high levels of endotoxin. So that's a compound that is released from the cell walls of certain bacteria, and it can cause an immune response. Or we might find that there are protein-based markers like zonulin. So that's elevated. That's an indirect marker that the tight junction proteins are not functioning properly. And then there are also other tests we can do using specific substrates that a person would drink. And then we would measure the amount that is in their urine versus in their bloodstream, and then determine how does that compare to a healthy individual? How does that compare to the ranges that we would expect to find if they had normal levels of intestinal permeability? So there are a lot of different ways that we can measure that. And it is something that is observed in the research setting. Now, does that mean that every person who has loose bowel movements or gas or bloating or brain fog also has leaky gut? No. And for a person to just say, you have symptoms, like you have brain fog, so you must have leaky gut, that's not accurate. And actually, there really are no outward symptoms of altered intestinal permeability. There's no specific gut microbiome profile. There are no GI associated symptoms that are known to be caused by leaky gut. It's one of these phenomenon that are kind of fly under the radar. Leaky gut is not a disease. It's not something that can be diagnosed or something that is treated. It certainly can be something that coincides with another disease, like I mentioned with IBD, and could potentially resolve if IBD is managed. But again, it's not that we have a medication to specifically address the intestinal permeability.
Mm-hmm. Perfect explanation. Thank you. You have really vast knowledge in this regard. Gabrielle, about the probiotics, can we move to this topic?
Yeah, absolutely.
Okay. Are probiotics supplements effective, or is it better to eat fermented foods, for example, instead of that?
Oh, that's such a good question, because you can kind of like go and talk about two different things. So as far as probiotics go, as I mentioned, probiotics do have specific applications, but the issue is that we can't just take any old probiotic or any old blend, like we do with a multivitamin, just take it every day and like, then we're all set. Most of the evidence indicates that the effects of probiotics are strain or subspecies specific. So we have the species, and then we have a subspecies. Like we can compare like dogs and dingoes. So like if you want to have a family pet, you want to have a dog, you don't want to have a dingo. So it's the same thing with a probiotic. You want to have the right probiotic for the job. If you have diarrhea versus having constipation versus having abdominal pain, you may be taking 3 different probiotics, one for each one of those issues. And also there's still not a strong consensus on the duration that's required or the dosage. There's probably some strain specific dosage needs as well. There's no clear indication that having multiple strains is better than single strains. In some cases, single strains actually appear to be more effective. But again, we also have the issue of having relatively low amounts of replication with these studies. There are really just a few probiotic strains that have been studied by a number of different research groups, and that the findings have really been replicated enough to say, with confidence. Like, for example, S. boulardii. S. boulardii seems to be very effective for reducing antibiotic-associated diarrhea. So again, it's one of those things that we really need to, if we want to make the most use of our investment, if we want to, spend our money wisely, then we want to be taking probiotics in a scientific way and not just like, whatever probiotic every day. Now, in terms of fermented foods, so fermented foods have been like microbes have been added to these foods, and it was used to be done to just preserve shelf life. But now we found that there are some fermented foods that could potentially have health benefits because of the microbes and because of the postbiotics or the products that they are creating as they are fermenting the carbohydrates in that food. So fermented dairy, like yogurts and kefir, are associated with improved cardiometabolic health. And so that's actually the only probiotic food. That's the only food that can carry the label of being probiotic, because it adheres to the current World Health Organization definition of probiotics, that are live microorganisms that, when ingested, confer some benefit to the host. When ingested in adequate amounts, confer some benefit to the host. So dairy fits that. But other fermented foods don't yet fit that definition. So it's either they haven't been studied enough, or that they have been studied a little bit, but they don't appear to actually improve health. Now, there was a study done fairly recently where they took 2 groups. They had one group increase their dietary fiber intake, and they had another group increase their fermented food intake. And this really made headlines, because everyone was like, oh my gosh, the fermented food group saw this reduction in immune response, and they saw this marked increase in diversity. Well, there are a couple things to remember. Anytime we eat food, we are ingesting the microbes that are in that food. And those microbes will then be measurable, they'll be identifiable in our stool sample. So if you eat a lot of yeast-containing foods, you'll have more of those specific types of yeast in your stool sample. That doesn't mean that those microbes have actually taken up residence in your GI tract. They could just go in one end and out the other, because that's what they do, because they actually haven't, they haven't taken up residence, but they'll show up in your stool sample just like your native microbes. The other thing to keep in mind is that when we want to determine like if something has improved our health, we want to look at like meaningful metrics. And the issue that I had with the way that this study was promoted is that it was kind of provided like without context, the information about the inflammatory markers. And so when I actually looked at the magnitude of change in those inflammatory markers, what I found was that in both groups, they were healthy at baseline. So they had normal levels of inflammatory markers at baseline. And then after the intervention, they still had normal markers. They still had normal levels of inflammatory markers. So they didn't really improve their health. They maintained their health, which is great. But then we would say both groups maintained their health. And so it's, whether you have fermented foods or dietary fiber, you didn't have an issue. That's kind of the problem there. Like had the group started with elevated markers and said like, okay, they have some increased level of inflammation that could be problematic, and then the fermented foods would significantly reduce those markers and fiber didn't, then that could be, something to write home about. But I personally just thought that was a little bit of a it was being oversold, the effects. And of course we didn't have a control group either. So there was nothing to say, like, what about if we just don't do anything? Like, do these inflammatory markers change? And does microbial diversity change over time? So just some things to think about. Now there's, if you wanted to eat fermented foods, there's certainly a chance that you could pick up some of the microbes that are in those foods, and they might enrich the population in your gut. But not all of the microbes that are used in food production would want to move into the environment in your gut. So it's not like every microbe out there is going to just be able to live in your intestinal tract and contribute to the microbiome in a meaningful way, which is good because we wouldn't want that to be the case. We wouldn't want to be so easily influenced by everything that we're eating, because that would mean that we would be very susceptible to pathogenic infections.
Perfect. So we should consume enough fiber for sure. Maybe some kefir sometimes.
Sure,
and should a healthy person use the probiotics supplement? Or, your recommendation, just simply said for normal person.
Right. Yeah. So it's kind of funny because people, you could look at some of the literature on healthy people taking a probiotic and say like, oh, how disappointing, like nothing happened after they took this probiotic. And it's like, well, no, I mean, that's not surprising. And we do need to have evidence that shows you do this thing and there's no effect. That's how, don't do that thing. So if you're a healthy individual, there is very little evidence that you'd be benefiting from taking a probiotic supplement. So the recommendation would be to instead use that money to, go grocery shopping, and buy the fruits and vegetables and whole grains and everything to support your native ecosystem, because things are running smoothly like they should. And so there's really no reason to add a probiotic. And even if you, and even like the types of the variety of diseases, I'll say that probiotics could benefit, it's still kind of a short list. So, having something like a headache, headaches or fatigue or sleep, sleep issues, the not to say that it's not going to exist, but there's not a probiotic for that yet. You're looking mostly at things like diarrhea, constipation, abdominal pain, and some management of some symptoms of IBS and IBD.
And as you mentioned, there is variety of the healthy microbiome in the world. And if you are a healthy person, why would you want to affect it when we know that there is not just one good thing or one bad thing? You know what I mean? Yes. So if it works, it works. Okay. Well, and let's say I am not healthy. I have some condition. How to choose the right probiotics? Should I ask the doctor probably?
You could do that. There's also a really great resource out there, and I will actually be next week. I'll be hosting a webinar on probiotic supplementation as well. But
Perfect.
the resource is it's AE Pro Bio. So you can go to that website. They also have AE Pro Bio is the tag on Instagram. A group of researchers have put together a whole compendium of the research and potential applications of dozens of different probiotic strains and products. Now, I don't obviously back any specific product or anything like that.
Of course.
But it is a really helpful resource to at least be able to go in and say, okay, it looks like, if I'm worried about, traveler's diarrhea, I'm going to be going out of the country and whatnot, and I want to just, have an extra layer of protection, then I can look on and say, oh, okay, it looks like these few strains have been used in studies on traveler's diarrhea, and they all have, like a grading of a level 1 grading of evidence. So that's like high quality evidence. This came from a randomized control trial. It looks like it's been replicated. This seems to be the most reasonable course of action if I want to take a probiotic, or if I'm going to be going on antibiotics or something like that. So that's where I would recommend to start, and and you can make the most prudent decision, I think, that way.
But look, as we are all different in the microbiome way, let's say, then the possible, well, I'm not sure if it's possible, but the best scenario would be some personalized mix of probiotics for each person, right? But I guess we cannot even know what would be the mix if we could do it, right?
Right. Because
what
we would, so anytime we have an intervention, we need to know like where we are, what are we aiming for, right? So if we have a personalized list of probiotics, that means that we need to know what this person has a deficit in or has an excess of. And to know that, we have to have a reference range, right? We have to know like this is what we would want to see, in a healthy person. And I wish I could remember. The reference, but I've mentioned this in a couple previous talks that there was a Belgian research group. It could have been. I think the last name is Backhead. I could have been out of that group, but anyway, they said that they would need something like 80,000 samples to be able to start determining what could be considered a healthy microbiome profile, like a reference profile. And that would be 80,000 samples from, people from all over the world. And it might be that we would have to stratify that like by ethnicity, by gender, by age. So in kind of the same way we do with certain blood markers, like if we're looking at, creatinine or something, or, one of the markers for like kidney function, glomerular filtration, that we have different reference ranges based on race. And so we still don't have that. We're not anywhere close to that level of sophistication in being able to determine like what we would expect to see in a person and where we would want to aim. So to say like going towards personalized, probiotics or even, personalized dietary interventions, that's putting the cart so far before the horse.
It's crazy how the charlatans are ahead of time, right? In the research of gut microbiome.
They're creative for sure.
Yeah, something like that, I would say. Okay, is there any prevention in regard to intestinal cancer or colon cancer?
Yeah. So colorectal cancer. So I had mentioned previously that the most realistic means of prevention would be really through diet and lifestyle factors. So making sure that we're having that diet that provides adequate fruits, vegetables, whole grains, legumes, nuts, and seeds, and that we limit our intake of processed meat products. There's some emerging evidence that the ratio of dietary protein to dietary fiber may also play a role, because there are certain microbes who can ferment peptides, so proteolytic fermentation, so the little like kind of smaller units of proteins. Not all microbes are happy to do that, so we'll lose a lot of diversity if we're eating like a meat only diet. And as they're producing energy from those peptides, they're producing some potential carcinogens like p-cresol, for example. So if we have tons and tons of protein and inadequate or no fiber, the microbes that can live on proteins, on peptides, will go ahead and do that. But then they're potentially also producing these carcinogenic compounds. Whereas if we have, a moderate protein diet, but we're also eating adequate fiber, then we're going to be still providing those microbes with those carbohydrates that are kind of reserved for them because they pass through our small intestine undigested and they get to the large intestine. So those will be available, and most of them would use those carbohydrates preferentially over the amino acids. And so we might not produce as much of those carcinogenic compounds. And then other lifestyle factors, smoking, alcohol use. It's kind of all like the boring, prudent stuff that, isn't too fancy or anything, but, often does come back to the basics.
Yeah, basics, always. Okay, how about artificial sweeteners? Do they damage gut health microbiome?
Mm-hmm. So if you
gut
are...
microbiome.
Yeah. If you are a rodent and you are ingesting super physiological doses of the artificial sweeteners in their pure undiluted form, which we don't do as humans, then there's some evidence that yes, you could develop some insulin resistance, some metabolic perturbations. In humans, though, there's really the evidence suggesting that artificial sweeteners have any effect on the gut microbiome or gut-derived hormones is either limited or just non-existent. So like, for example, with aspartame, they have fed humans aspartame and they found that there's no effect on the gut microbiome or on gut-derived hormones.
Dosage makes poison.
Right. Yes, exactly. Yes. And it would be almost impossible probably for us to be able to ingest enough of any of the artificial sweeteners. So for example, like with sucralose, when we use sucralose, when we buy it, we're getting sucralose mixed with maltodextrin or dextrose. I can't remember which one, but in any case, because sucralose is 600 times sweeter than sucrose, it would be very hard to have a usable amount of it. You'd need to have 1 granule of sucrose and put that into your coffee, and it would be like mind-blowingly sweet. And so they cut it with maltodextrin. So you're getting actually more maltodextrin than you are of the sucralose. So your actual dosage of sucralose is extremely low, and that's not what they're doing with rodent studies. So the dosage definitely makes a huge difference. But also, the compounds are not going to interact with the microbes. Like, it's not something that they're going to be able to metabolize, and it's not something that has antimicrobial properties. Then it's going to be pretty inert as far as the microbiome goes. It's going to just kind of, go in one end and out the other, and the microbes will just do their thing around it. So that's probably what's going on with these artificial sweeteners that don't appear to have any effect on the microbiome. There are some that are not artificial, but are non-nutritive, like sugar alcohols. Many of those are prebiotic, and so they can lead to, gas and bloating. So you could potentially see them as like beneficial a little bit, like maltitol. But they, obviously, or sorbitol, they cause a lot of GI distress. So it's probably more stressing to us than it is to the microbiome to have sugar alcohols.
In fact, correct me if I'm wrong, but people always solve the thing like, this affects my microbiome, but everything affects your microbiome. But it doesn't mean it is in a bad way, right?
Yeah, exactly. Potentially anything that you ingest can have an impact on the microbiome. Even your water, your tap water could have an impact because, again, you are ingesting microbes in the tap water. It would probably be more accurate to say, like, we could probably measure changes in your fecal microbiome, like in a stool sample. And that would probably we would probably see like more significant changes there than we would actually see in the microbiome itself and like the totality of the microbiome of your whole GI tract, because we're looking at, about 40 trillion microbes there and about 220,000 genes. So like if we ingest even a probiotic, that taking 1 million microbes is like a drop in the ocean. So it's probably more likely that we could say like, yes, you'll probably be able to measure these in your stool sample because they went in one end and out the other. but was there an actual change in the taxonomy of your microbiome? So did who's there change? And then was there a change in the function of your microbiome? So even if, some of the members of the population changed, did that actually change what the microbiome is doing as this sort of collective? And so I think those are the functional question is potentially more important to answer. But it's one that we haven't been able to answer as easily as the taxonomic question. We've been able to do that for quite a long time. So it's just kind of also waiting for the technology to catch up, to be able to analyze it in different ways, and with really fine resolution, and then to determine from there, okay, and then what does this mean for the health or disease of the host?
Sounds very interesting. I'm looking forward to it if I will live in that time. How about insulin and sweeteners? There are opinions that if you use artificial sweeteners, it affects your insulin, even though there is not a rise in blood sugar.
Right. Yes. So, and I think that's probably looking more at the peripheral effects, even the effects on the central nervous system and whatnot. You can see a rise in performance after swishing Gatorade in your mouth. You're not actually absorbing those nutrients, but there is some impact on performance. So, and in the study that I had mentioned, in one of the studies that I was mentioning about sucralose, they did find that in a very small subpopulation of humans who were ingesting the maximum amount of sucralose, considered safe for human consumption, I think there were 7 individuals that it did experience some perturbations in their insulin sensitivity. But that was out of, a much larger group. So is that indicative of, does that show that there's actually a cause and effect relationship? Not necessarily. And, when they used their stool samples to transplant that into rodent models, the study design used something called pseudo-replication. So they put, one person's stool sample into multiple rodents. And that can often amplify the number of samples that you have in sort of an artificial way. And so I think that's where a lot of that might come from is this, the way that they've used fecal transplants to determine whether they can replicate this insulin resistance in rodents and ostensibly determine a cause and effect relationship. So probably remains to be seen. There could certainly be an inter-individual variability with how artificial sweeteners could influence someone's insulin sensitivity. But also, if we're looking at a less controlled sample, is a person having artificial sweeteners and, they're having they've switched to diet sodas. And so now they're more liberal with the rest of their diet. And maybe they have actually, increased their adipose tissue and then, had a reduction in insulin sensitivity, not caused by the artificial sweeteners, because there's really no evidence that's going to cause weight gain or insulin resistance, but because of these other factors. Yeah,
correlation is not causality. Right. Great explanation again. About fecal transplants, as you said, do these work? I know we did not have this in questions, but I'm just curious.
Sure, yeah. So fecal transplants right now are in the U.S. They're only they have a special use approval by the FDA, so they can be used to treat C. difficile infections that are resistant to antibiotics. So a person's tried a few different antibiotics, they still have C. difficile. At that point, then they could undergo a fecal transplant. So fecal transplant is just like it sounds. We're taking fecal contents from a healthy donor and providing them to a person with a disease, either through enema or through encapsulation. It is really, really effective for treating antibiotic-resistant C. diff. It hasn't been as effective or promising for inflammatory bowel disease or for modifying body weight. It seems to have no impact on body weight in humans, even though there is definitely an impact in rodents. And it doesn't appear to be very consistently helpful in inflammatory bowel disease, and in some cases actually has exacerbated the disease. So it's not really, right now the applications are very limited. But it is something that's used in research, to try to replicate a specific phenotype, so a specific disease or a specific body weight or behavior. in germ-free rodents. So they have one rodent that is raised without a microbiome at all, and then they take a fecal sample from a specific type of rodent and put it into that germ-free rodent, and then they watch what happens. Now, the issue with that is that it is about 98% effective, which is actually highly unlikely, or there's some sort of publication bias. So, because to say that works in 98% of cases is just kind of statistical improbability. So there is that limitation, with the way that that shows up in the literature, but it is it's helpful in terms of like identifying mechanisms and getting close to establishing cause and effect in rodents. But again, that's not indicative of cause and effect in humans.
Perfect. By the way, if you're watching South Park, this topic was there also. I was crying and laughing at the same time.
I know, wasn't it like Tom Brady's like fecal sample or something?
Yeah, exactly. So we won't
athlete.
lose weight in real life if we do fecal transplants. That's too sad.
I know.
Okay. How can we fix our microbiome after taking antibiotics?
Yeah, we might not necessarily need to fix anything. It really depends. I mean, there are some antibiotics that have pretty minimal impact on the microbiome as a whole. Of course, there are some that will have a lasting impact. You might have a reduction in diversity or certain taxa for 5 months or longer. So there's not, again, a lot of research on kind of like rebuilding the microbiome. But if you are experiencing, like the antibiotic associated diarrhea, probiotics would be a helpful application in that respect. Not necessarily that they're rebuilding the microbiome, but they are at least reducing one of the, side effects of antibiotics. Based on the very limited amount of evidence that we have, it looks like it's helpful to just go back to what you were doing, if what you were doing included eating things like, fruits and vegetables, whole grains, and getting some physical activity. If that's not what you were doing, it's a great time to start because that's one way that you could enhance the potentially enhance microbial diversity. Or if you were doing those things beforehand and you wanted to get back to that place once again, then you would give yourself an autologous fecal transplant. But don't do that without a doctor. But that is another thing that's worked very quickly, is that when people say they know they're going to go on antibiotics, so they take their own fecal sample beforehand and then they provide it, to themselves afterwards, then that's helpful. Interestingly, they did find that taking a lactobacillus predominant probiotic actually delayed the reestablishment of the prior level of diversity. So it was like after using antibiotics, there was an opportunity for the probiotic lactobacillus strains to sort of overpopulate and potentially outcompete some of the native microbes. And so there was a sort of a version of dysbiosis or imbalance that occurred with that. So another thing to potentially keep in mind, that, it's like taking probiotics isn't always the answer. And if you don't have any antibiotic associated diarrhea, then it's probably prudent to just, eat fruits, vegetables, and whole grains and all that good stuff. And then, get some aerobic physical activity in there.
Perfect. We had a question about, does training, walking affect gut microbiome? You already said that aerobic activity does, but do strength training affect this also?
We don't know that yet, but I am involved in a collaboration that's looking at the effects of a bout of resistance training on GI distress and markers of intestinal permeability. And we're also identifying whether there are some gender-specific associations between microbiome and these markers of GI distress and permeability. And so we're going to kind of be hopefully at the forefront of some of those answers, because most of the literature really is on endurance athletes. Or like sport, or like, recreational athletes. So they're doing a combination of aerobic and resistance training. And even from that literature, I believe that the most recent systematic review found that only 4 of the studies had controlled for diet separately from exercise, and only 2 of them found that there was a direct potential effect of exercise. So we're still sort of like wondering, how much of the diversity is associated with the diets of these people versus how much is really associated with exercise.
And many people who are athletes or who are trying to lose weight do this and also change their diet. So yes, that's quite interesting question. I'm looking forward to this also. Gut-brain axis. Is it a real thing? Could our microbiome influence our mood levels of neurotransmitters in brain?
It is a real thing. So there is a bidirectional communication through the vagus nerve. So that's kind of a direct highway between the brain and the GI tract. It controls about 75% of our parasympathetic nervous system activity. So that rest and digest, the rest and digest sort of set of activities that we would be going through after a meal. And then there's also plenty of evidence that microbes are producing precursors to neurotransmitters or neurotransmitters themselves that could act in the periphery, or they could potentially either cross the blood-brain barrier or enter via some of the structures that aren't protected by the blood-brain barrier. Now, again, we don't have a cause and effect relationship, and most of the research looking at the relationship between behavior and the gut has been done in rodents, or it's been done with, with imaging, with brain imaging in humans. And so we're still really in the phase of determining potential mechanisms there and sort of proof of concept. So to say that like your microbes are going to affect your mood wouldn't be substantiated at this point, but there is certainly a potential that they could influence mood in the same way that they influence drug metabolism. And so there's an individuality to that as well. But we just don't really know how and to what extent.
Of course. Well, could we give some at least basic recommendations to people with ulcers or Crohn about gut microbiome? Do he have some implications from the research that could help them?
Sure. I wouldn't say that we have really actionable recommendations yet, but there are emerging patterns when we're looking at, so for example, when we take these, when we're sequencing the microbiomes of a bunch of different people with ulcerative colitis, like from different studies in different areas in the world, or with Crohn's, and we compile all that together, we can start to see that there are certain patterns. And we can do this with a number of different diseases, that in some cases, like with colorectal cancer, for example, we see that there is an enrichment of pathogens. And in other cases, we might see that the number of pathogens is normal, but there is a lower number compared to the healthy controls of beneficial microbes. But there are a lot of microbes that are just shared, but because we're human. And so we have to be very careful not to draw a spurious correlation, accidentally say like, oh, this microbe must play a role in this disease activity, because it could be just that microbe is enriched in that specific group of people because of their geographic location, or because of their age, or because of their gender. So it's still an area where we haven't been able to identify any cause and effect relationships, but there is an indication that the microbes could play a role in the disease progression, and also that the disease progression could influence the microbial community, because we are modifying the environment in which they exist, and we're modifying the immune activity. And then also there's the impact of, various drugs that people might be taking to modify, to manage their disease. So most we have, potential correlations or relationships, but still trying to parse out which of those are actually meaningful. And so again, it's about, working with their doctor to find the treatment plan that's best suited for them, and having the potential in some cases for probiotic supplementations added on. But again, that's going to be, just complementary, if anything.
Okay, and look, I would like to transfer to some of coaching topic, if you don't mind.
Yeah.
I believe that psychological aspect of coaching is very important. One of the main thing of coaching. Look, I know you have a very, very good approach in this regard. Could you tell us something about how you work with clients, in this regard?
Yeah,
Thank you.
absolutely. And I appreciate the kind words. So I have been working with one of my good friends and colleagues, Shannon Beer, for a few years now on what we call the comprehensive coaching framework. And it started when we noticed that there were a lot of potentially harmful messages coming from the fitness community that placed a lot of blame and created a lot of shame in clients who were repeat dieters, who would use a method like macros, for example, to successfully, as it were, to successfully lose weight, and then they would regain it. And then this was this idea that, if you regained the weight or you had a difficult time losing weight, that you weren't motivated enough, that you didn't want it bad enough, that you didn't have enough self-control. And it really creates sort of repeat customers because they are being misled about what it is that leads to weight loss and the maintenance of weight loss and even the probability of maintaining more than a very small % of weight loss long-term. And it's not to say that it's impossible to lose body weight and maintain weight loss long-term. But I think we really need to be more transparent about the realities of that process and look at it as like a contact sport where a person has to be able to provide informed consent to do something that could potentially be harmful for them. And although we may not see the harm externally, there can certainly be psychological harm. And we also wanted to approach our clients in a different way that was less prescriptive, less of the sort of like, I'm the expert on everything and I'm going to tell you what to do, because we found that that isn't actually helpful for long-term skill building, long-term and long-term maintenance. So what helps people actually decide, what is important to them and make meaningful changes, it actually comes down to respecting their autonomy and helping them to feel and build their competence, their self-efficacy, and to actually collaborate with them. And so we were trained in motivational interviewing and acceptance and commitment training and cognitive behavioral coaching. And we're also adding some other aspects to the framework as well that essentially is aimed at coming together with a client to collaborate. So the coach is the expert in certain areas, and the client is the expert on themselves. And we actually need one another to produce this alliance and work together, a lot like going on a trip with a travel guide. So the coach is the travel guide. The coach is the expert on nutrition and training and how to have a productive conversation that draws out the client's own motivations and the client's own reasons for and own commitment to making a change. So we're talking about the possibility of making a change that's actually meaningful to the client because we've been able to draw out why it is they want to meet that goal. And in many cases, helping them realize that the reasons that they want to meet that goal aren't necessarily going to be met by that goal. For example, getting a six-pack does not necessarily improve your body image. It just makes your body look more attractive to you, so you like it more. But it doesn't mean that if you were to regain the weight that you would maintain that positive body image. So your body image hasn't changed. You've just changed your body. If you want to improve your body image, you can do that via body image work so that regardless of whether you have a six-pack or not, you aren't criticizing yourself and making yourself have a bad day. And then over the course of working together, we're helping to enhance their psychological flexibility, their ability to cope with just the everyday challenges that come with, pursuing a goal and making decisions aligned with their values. So it might be that we're helping them reframe thought patterns that kind of keep them stuck in cycles, or we're helping them to take values aligned actions, even in a stressful situation. And that the overall outcome is more than just a physical change. It's actually about flourishing health. So it is prospering in the areas of physical, social, psychological, and philosophical health. And that we're working with them to keep those things in balance so that they're not losing out on mental or social health because they're in pursuit of this really extreme physical goal. So it is not to say that we're like reinventing the wheel. It's really just that we've taken a lot of the best practices from different areas that we've borrowed from like a therapeutic way of helping people change. And we brought those to the coaching side of fitness and helped to make them more actionable so that coaches can play a different role in the lives of their clients. And it becomes less about, providing them with one specific skill to get to just this one specific outcome. And then once they reach that, that goal weight, then it's done. No, we're here to help them feel much more capable and to reach a bunch of different goals. And so they can continue working with us, whether they have a weight specific or a weight neutral goal, and that the responsibility and the credit for change lies with the client. And we are no longer responsible for the specific outcome, which is actually beneficial for both of us, because that means that we don't have to worry that like our livelihood is based on the outcome that this client reaches, because their behavior is actually outside of our control. What's within our control is our behavior. What's within our control is the way that we engage with the client. And so we are providing them with the service of an experience. And then the client is in charge of their outcome. And that actually is what's going to support their long-term motivation, because that's about their autonomy, their choices, and their level of competence, that they know that they did this. They came up with these great ideas, and we collaborated together, and then they did this thing. And they know that they're capable of much more, and that they don't need us, but that we are hugely helpful in supplementing their learning journey. So it can support really long-term relationships that are much more fulfilling for both people.
That's perfect. Really comprehensive approach. Dr. Fundaro, would you recommend something generally to coaches that they could do better from what you've seen, how coaches work with their clients? Because you have many experience. So
yeah. I would say maybe not necessarily to do better, but I think one helpful shift in state of mind based on the coaches that I've worked with. I think that a lot of coaches do have a fear. I think a lot of coaches do take responsibility for the client's outcomes quite a lot, that they think that the clients, that they will only be employed if their clients reach this specific set of results. And that's part of kind of the culture of the industry. And so when you come together with a client and you expect that, and they expect that, that puts almost all of the pressure on you. Now they're looking at you to motivate them, to hold them accountable, to tell them exactly what to do, to come up with all of the solutions. And then you're put in this place of, I have to know absolutely everything about everything. And I have to tell them the right thing to do. And they have to get this specific result, because if I don't, then I've failed them, and then they're going to leave. And that is like, what a way to burn yourself out. So shifting the expectations and shifting the relationship to that collaboration, where I am here to be the expert on a conversation that is likely to help you make changes long term. And then I'm here to provide you with the skills and tools and information that will supplement the journey that you want to go on. And you, as the client, are responsible for your results. You get all the credit for that. And that way, I know that with the proper training and experience, I can provide that quality level of experience for the client. I can direct a conversation appropriately. I can provide recommendations, with their permission in a collaborative way. And I can say, well, here's this exercise that might be helpful. And then they can choose to use it or not. And it's completely up to them. They're still in charge of, the application and of reaching their goals. So if something doesn't go to plan, rather than you feeling like you failed and you need to provide all of the solutions, you can say, what do you think was going on there? And then the client says, I think it might have been this and that. And you say, oh, okay. It sounds like you kind of have an inclination and you want to do some things differently next time. And they say, yeah, exactly. And then you have this conversation about what you do differently next time. And it is still that you are collaborating, but the outcome is still their responsibility. and you have done what they expected, which is you had this really helpful, evocative conversation with them. And then they've done what they expected, maybe or not, but there's no shame, there's no blame if they don't. And so you also are creating this really safe, facilitative atmosphere where you can experiment and find like what actually does work for them long term, rather than trying to like get them into this cookie cutter place. And it's so much more beneficial for the coaches as well, because now you don't have to be, I mean, if you want to be a macros coach and do nothing else, that's totally fine. But like you could be a macros coach and also all of these other things. And so you can really, work with a wider variety of clients too.
Yeah. I personally use flexible dieting, but I also, I'm trying to convert client at the end of coaching to intuitive eating, to use what he learned without some applications, so waiting food, etc. Because we remember the prices of food. It changes with, discounts, etc. But the calories are the same, and you need to just know approximately how much money you have to buy it, how much calories you burn, etc. And I think it could work well with some people who like this kind of approach, of course.
Yeah, that's such a great analogy.
Thank you very much. Gabrielle, look, I'm aware of the fact that our time is limited. Could we have last 2 questions? Okay. First is a very important question. How much you can deadlift?
Oh, gosh, I haven't deadlifted in a long time. I would say, I think my last deadlift was 305 pounds. So
wow.
I can't with that 150 or so kilos, a little bit less than
something
that.
Like that. How much you weigh? How tall are you?
I'm 5 feet tall, and I was 63 kilos for that.
Respect, really. That's really good. Okay. And the last question, I always ask people who are guests here, if you could give one tip to everyone on earth in your specialization, let's say it is gut health for now, what would you tell the people?
Oh, wow. So one gut health specific recommendation for everyone on the planet.
Yeah. Or any recommendation if you choose to.
Okay. I would say I'll give 2 recommendations. So the first recommendation for gut health specific would be try to eat plants at most meals. Just very, because a lot of people aren't doing that. I think that the second recommendation would be to ask if things feel like they're not going well for you in your life, and this isn't in a blaming type of way, but like, and I guess in any case, I think it's helpful to check in with yourself and ask like, why did I choose to do that thing? Just in a curious way, not in a blaming way, but like what was going on for me there? And that can certainly be applied to nutrition and fitness as well. Just starting to develop that self-awareness of like, what is driving my behaviors? And that is where we can start to potentially change things. When we just get in touch with ourselves, we just accept like, this is why I do this thing. That's when we can start to change if we want to.
That's perfect, because we are here now, not just by coincidence. It is because of our past actions and choices we made. So I think it is a good tip. Okay. Is there anything I've missed we should say today?
That was fantastic. I know. I think we covered so much, and people know where to find me, and so it was awesome. Thank you so much for having me.
Okay. Thank you very much. Also, thank you for your time, Dr. Fundaro. We are honored to have you with us today. And farewell to all you guys. Next guest should be Dr. Karl Nadolski about endocrinology, and maybe we'll see Tom Brady also. Just kidding. Okay. Thank you very much for your time, and have a fine, evening.
Thank you. Thank you so much.
Bye-bye.
Bye.